Hi, my name is Olivia, and I am presenting on the effects of polyunsaturated fats on inflammation. Recently, there has been a bit of a war on seed oils across many media platforms. Anti-seed oil influencers make claims that seed oils contribute to inflammation, obesity, and other chronic diseases, and have encouraged many to eliminate these oils from their diets altogether. Seed oils are cooking oils that come from various plants and are also known as vegetable oils. There are 8 common ones referred to as the Hateful Eight by many health influencers. They include canola oil, corn oil, cottonseed oil, sunflower oil, safflower oil, soybean oil, grapeseed oil, and rice bran. These oils are polyunsaturated fatty acids, which is actually a type of fatty acid with 2 or more double bonds. They are critical for brain function, blood pressure regulation, immune function, cell membrane structure, and cardiovascular health. There are several types of polyunsaturated fats, but 2 that are considered essential nutrients as our bodies do not synthesize them, and therefore we need to obtain them through food sources. These are omega-3 and omega-6 polyunsaturated fatty acids. These are also known as alpha-linoleic acid and linoleic acid, respectively. A diet consisting of a balance of these 2 PUFAs, or polyunsaturated fatty acids, is actually considered healthy. So I wanted to set out to discover if these vilified seed oils, also known as polyunsaturated fatty acids, really do contribute to inflammation as much as these health influencers make it seem. So there are 2 questions I would like for you to consider as we navigate through this presentation. 1, are seed oils an ingredient that you avoid? Why or why not? And 2, what are ways that you can incorporate more omega-3 and omega-6 polyunsaturated fatty acids into your diet? So to answer my research question, I conducted a sort of mini literature review. I used keywords like polyunsaturated fatty acids, seed oils, inflammation, obesity, and chronic disease. Online databases such as NIH and PubMed were used to search for articles, along with other platforms such as the Mass General Hospital website. Inclusion criteria for the search included articles written about omega-6 and omega-3 polyunsaturated fatty acids, and more specifically, 8 of the most commonly used polyunsaturated fatty acids, and topics surrounding the ways in which these fatty acids affected inflammation and chronic disease in humans. 3 primary research articles were selected and studied in depth to determine the effects that polyunsaturated fatty acids have on inflammation. So the first article selected was titled Effect of Different Types of Dietary Fatty Acids on Subclinical Inflammation in Humans. This article sought to test the effect that replacing a diet in saturated fatty acids, or SAFAs, with a diet in polyunsaturated fatty acids, or PUFAs, has on systemic inflammation. This was tested through a randomized controlled crossover study with 15 overweight or obese postmenopausal women. There were 2 3-week dietary periods, one of which supplied participants with a diet high in saturated fatty acids, with a 1-week washout period, and 1 3-week dietary period during which participants were supplied with a diet high in polyunsaturated fatty acids, followed by a washout period. Metrics were recorded throughout the study. This study found that a diet high in saturated fatty acids led to an increase in LDL cholesterol, while a diet high in polyunsaturated fatty acids decreased levels of LDL cholesterol and C-reactive protein, which is a biomarker for inflammation. Interleukin-18, another biomarker for inflammation, showed no significant differences between the 2 diets. The second article was titled Effects of N6 Polyunsaturated Fatty Acids Compared with Saturated Fatty Acids on Liver Fat, Lipoproteins, and Inflammation in Abdominal Obesity, a randomized control trial. So this study sought out to test the effects of switching saturated fatty acids with vegetable polyunsaturated fatty acids on liver fat, inflammation, and metabolic disorders in the human body. 67 abdominally obese individuals were randomly assigned a 10-week isocaloric diet, either high in vegetable PUFAs or high in SAFAs with butter as the main source, without altering the macronutrient intake. This study found that when given the PUFA diet, participants experienced lower levels of liver fat. This study also indicates that PCSK9 levels, a biomarker for LDL cholesterol, were lower after consuming a polyunsaturated fatty acid diet. Additionally, biomarkers for inflammation, TNF receptors, and IL-1 receptors were lowered after consuming the polyunsaturated fatty acid diet as opposed to consuming the saturated fatty acid diet. The third article was titled "Exchanging Saturated Fatty Acids for N6 Polyunsaturated Fatty Acids in a Mixed Meal May Decrease Postprandial Lipidemia and Markers of Inflammation and Endothelial Activity in Overweight Men." This study sought to study the effects of a meal rich in saturated fatty acids versus the effects of a meal high in omega-6 polyunsaturated fatty acids on postprandial responses of overweight men who are at a risk of metabolic syndrome. This was studied through a randomized double-blind crossover design with 13 overweight men. All participants received 2 different interventions. After a 12-hour overnight fast, a blood sample was taken, and then 10 minutes later, each participant was requested to consume either a meal rich in butterfat or a meal rich in sunflower oil, both containing 50 grams of fat. Blood samples were then collected at a specific time intervals after consumption. Key findings included that the exchanging saturated fatty acids from butterfat for omega-6 polyunsaturated fatty acids in a mixed meal can decrease postprandial lipidemia and inflammatory biomarkers IL-6, TNF-alpha, and TNF receptors 1 and 2. So seed oils are omega-6 polyunsaturated fatty acids being blamed for the obesity epidemic and chronic inflammation experienced today. Maintaining a healthy ratio of omega-6 and omega-3 PUFAs is vital to preventing chronic inflammation, as too much of anything is bad. Omega-6 is being vilified because it is often used to make ultra-processed fast food that also happens to contain high amounts of sodium, added sugar, and saturated fat. And yet, omega-6 PUFAs are being blamed for all of the problems that come with consuming excess of those ingredients. According to the articles, omega-6 PUFAs actually do not contribute to chronic inflammation, as the biomarkers consistently showed decreased levels when participants consumed a diet high in PUFAs rather than saturated fatty acids. To answer my original research question, what is the effect of polyunsaturated fatty acid PUFAs on inflammation? I would say that across all the studies included here, decreased levels of inflammatory biomarkers were found when participants consumed a high omega-6 polyunsaturated fatty acid diet as opposed to a high saturated fatty acid diet. Consistent decreased levels of interleukins, C-reactive proteins, and TNF-alpha receptors across all studies indicate an anti-inflammatory effect on the omega-6 PUFAs, also known as seed oils. LDL cholesterol levels also decreased when a PUFA diet was consumed. So, in conclusion, when omega-6 polyunsaturated fatty acids are not consumed through ultra-processed foods in excess, they can actually have an anti-inflammatory effect on the human body. Here's my bibliography, and thank you so much for listening.